Next point, we are going to start with the de novo synthesis of fatty acids, that is lipogenesis. De novo means new synthesis. So we will learn how these fatty acids are synthesized using acetyl coenzyme A. In humans, fatty acid synthesis occurs mainly in the liver and lactating mammary glands, and also to a lesser extent in adipose tissue, kidney and brain. Fatty acid synthesis occurs in three stages. First is transport of acetyl coenzyme A from mitochondria to cytosol. Second, carboxylation of acetyl coenzyme A to malonyl coenzyme A. Third, reactions of fatty acid synthase complex. Now we will start with the stage 1, that is transport of acetyl coenzyme A from mitochondria to cytosol. Fatty acids are synthesized in the cytosol, whereas acetyl coenzyme A is formed in the mitochondria. Hence, acetyl coenzyme A must be transferred from mitochondria to the cytosol. Since acetyl coenzyme A is impermeable to the inner mitochondrial membrane, it is transferred in the form of citrate from mitochondrial matrix to the cytosol as shown in the above diagram. Step 1. Citrate is formed in the mitochondrial matrix by the condensation of acetyl coenzyme A with oxaloacetate, which is the first reaction in the citric acid cycle. Step 2. Then citrate is transported to the cytosol by enzyme translocase, where it is cleaved by the citrate lyase to oxaloacetate and acetyl coenzyme A. Oxaloacetate formed in the reaction must be returned to mitochondria. The inner mitochondrial membrane is impermeable to oxaloacetate. Therefore, oxaloacetate is reduced to enzyme 2-malate by the enzyme malate dehydrogenase, which is cytosolic. Then malate is oxidatively decarboxylated to pyruvate by NADP-linked malic enzyme. NADPH and carbon dioxide are generated in this reaction. Both of them are utilized for fatty acid synthesis. Then pyruvate formed in the reaction readily diffuses into mitochondria, where it is carboxylated to oxaloacetate by pyruvate carboxylase. We'll continue to stage 2. It is carboxylation of acetyl coenzyme A to malonyl coenzyme A. Carboxylation of acetyl coenzyme A to malonyl coenzyme A is the initial and rate-limiting reaction in fatty acid synthesis. This reaction is catalyzed by an enzyme complex acetyl coenzyme A carboxylase that contains biotin and utilizes bicarbonate as a source of carbon dioxide in the presence of ATP. Stage 3. It is reactions of fatty acid synthase complex. Once malonyl coenzyme A is formed from acetyl coenzyme A, the de novo synthesis of palmitic acid is carried out in cytosol by fatty acid synthase complex. It is a multi-enzyme complex possessing 7 different enzymes and 1 acyl carrier protein molecule. The fatty acid synthase complex has 2 active SH groups. One SH group is of 4-phosphopantethene moiety of acyl carrier protein, and another SH group is of cysteine residue of enzyme ketoacyl synthase. Both SH groups participate in fatty acid biosynthesis. Fatty acid synthase complex is a dimer. Each monomer is identical, consisting of all 7 enzyme activities of fatty acid synthase and an acyl carrier protein. The 2 subunits associate in a head-to-tail arrangement so that the phosphopentethene SH group of one subunit and a cysteine SH group of another subunit are closely aligned. Though each monomer contains all the enzyme activities, but only the dimer is functionally active. This is because the functional unit consists of half of each subunit interacting with the complementary half of the other. The two functional subunits of fatty acid synthase can synthesize two fatty acids simultaneously. The fatty acid synthase complex catalyzes the following reaction. We'll go step by step. In the first reaction, catalyzed by acetyl transacylase, the acetyl group of acetyl coenzyme A is transferred to the cysteine SH group of the 3-ketoacyl synthase of fatty acid synthase complex, which is designated as E. In the second reaction, the malonyl group of malonyl coenzyme A is transferred to phosphopantethene SH group, that is pan-SH of acyl carrier protein catalyzed by malonyl transacylase to form acetyl malonyl enzyme. The acetyl and malonyl groups covalently bonded to SH groups of the synthase undergoing a condensation reaction. This reaction is catalyzed by 3-ketoacyl synthase. The acyl group attached to cysteine SH is transferred to malonyl group bound to SH pantethene. Simultaneously, the malonyl moiety loses carbon dioxide, forming 3-ketoacyl enzyme. The 3-ketoacyl enzyme undergoes reduction at the 3-keto group at the expense of NADPH as electron donor to form 3-hydroxyacyl enzyme catalyzed by 3-ketoacyl reductase. The 3-ketoacyl enzyme is dehydrated by 3-hydroxyacyl hydratase to give 2,3 unsaturated acyl enzyme. 2,3 unsaturated acyl enzyme is reduced to form acyl enzyme containing 4 carbons by the action of enoyl reductase, and NADPH is the electron donor. This acyl group containing 4 carbon is now transferred from pantethene SH group to cysteine SH group. To lengthen the chain by another 2 carbon unit, the sequence of reactions is repeated, a new malonyl residue being incorporated during each sequence, until a saturated 16-carbon acyl radical, that is palmitoyl, has been assembled. Thus, after a total of 7 such cycles, palmitoyl enzyme is formed, which is liberated from the enzyme complex by the activity of a 7th enzyme in the complex thioesterase, that is deacylase. Regulation of fatty acid synthesis. De novo fatty acid synthesis is regulated by acetyl coenzyme A carboxylase enzyme. Acetyl coenzyme A carboxylase, which catalyzes the formation of malonyl CoA that we have already studied. It is the rate limiting enzyme in the fatty acid synthesis. It is regulated by following ways. First, allosteric regulation, that is, acetyl coenzyme A carboxylase is allosterically activated by citrate and inhibited by its own product palmitoyl coenzyme A. Second, hormonal regulation. The activity of acetyl coenzyme A carboxylase is also controlled by hormones. Glucagon and epinephrine inactivates the enzyme. In contrast, insulin activates the enzyme. Thus, fatty acid synthesis is stimulated by insulin and inhibited by glucagon and epinephrine. Third, nutritional regulation. Synthesis of acetyl coenzyme A carboxylase is increased with high carbohydrate and low fat diet, which promote fatty acid synthesis. By contrast, synthesis of acetyl coenzyme A carboxylase decreases during starvation, diabetes mellitus, or high-fat diet.